Hello everyone, welcome to Teaching Pathshala. This is the second part of GPCR in which we are going to study about the cAMP pathway and IP3-DAG pathway. I have already uploaded the first part of GPCR in which we have talked about the basic things regarding the GPCR. So if you have not seen that video, I will mention the link in the description box as well as in the end of this video. I will also provide the link for the cell signaling playlist in which you are going to get all the different videos which has been uploaded till now regarding the topic of cell signaling. So if you are here for watching the part 2, I will request you to first watch the part 1 because all the basic and the necessary things has been discussed there, and this will not include the discussion of the basic things. In this part we are going to talk about the detailed pathway regarding the different GPCR receptor. So first you should go and watch the basic, that what is GPCR, what are the different factors that are playing important role in GPCR. So this concept should be clear, and after that you can watch this video. These all are the topics which we have completed till now. So in the previous video we have seen the introductory portion of the GPCR, and today we are going to see that in what different pathway this GPCR is involved. So today we are going to study about the cAMP pathway and IP3-DAG pathway. So the first topic of discussion is the cAMP pathway or cAMP pathway. So before moving to the pathway, you should know that what is cAMP or cAMP. So cAMP is a derivative of ATP. That means it is derived from ATP and participate in signal transduction by conveying the cAMP dependent pathway. cAMP or the cyclic AMP is a very important secondary messenger. And if we talk about the synthesis of cAMP, so cAMP is mainly synthesized from ATP in the presence of adenylyl cyclase enzyme. From the figure you can see that the structure of AMP and cAMP both are same, only the difference is that there is a presence of a cycle at the place where phosphate and the sugar is combining. So this is the only difference between the AMP and the cyclic AMP. When cyclic AMP used to degrade or decompose, it mainly result into the formation of a AMP, and this reaction will be catalyzed with the help of an enzyme phosphodiesterase. In the previous video, we have seen the function of the phosphodiesterase, that what it used to do and how it used to catalyze the reaction. In the previous video, we have seen one line in which it was written that the cAMP or the cyclic AMP is synthesized from ATP in the presence of a adenylyl cyclase enzyme. So first we have to know that what is adenylyl cyclase enzyme and what is the location or what is the role of this enzyme. So adenylyl cyclase is an enzyme that is located on the inner side of plasma membrane, and it used to catalyze the cyclization of ATP into the cAMP or cyclic AMP. So ATP is a adenosine triphosphate. So there is a presence of a 3-phosphate group in this structure. But whenever there is a presence of a adenylyl cyclase enzyme, at that time the pyrophosphate will release and one phosphate will remain attached to the overall structure, and that will result in the cyclization of a AMP into a cyclic AMP. In this way, the conversion of a ATP to cyclic AMP will take place in the presence of a adenylyl cyclase enzyme. Before moving to the main pathway, let's see what are the different molecules which used to enhance and inhibit the cAMP pathway. So the molecules like cholera toxin, caffeine, pertussis toxin, all these will enhance the cAMP pathway or activate the cAMP pathway. But if you talk about the inhibition of the cAMP pathway, this will include the cAMP phosphodiesterase enzyme that will lead to the inhibition of a cAMP pathway. And last is the GI protein, which is a part of a G protein but a inhibitory G protein, and that is why it is going to inhibit the adenylyl cyclase enzyme. So in this way there will be the less conversion or no conversion of a ATP to cAMP. So this is an important slide through which section B question can come, like which molecule used to activate the cAMP pathway, and they will give you a different option in which one option will be from this list only. So you should remember that which molecule is going to activate the cAMP pathway and which molecule going to inhibit the cAMP pathway. Now let's move to the cAMP pathway. Initially there is an absence of any kind of a ligand. So that's why there is an inactivation of a G protein. That means it will remain in a trimeric form and alpha subunit will remain attached to the GDP. So trimeric form is always an inactive form, and GDP is also playing a role in the inactiveness of this protein. But the situation changes whenever there is a presence of an external ligand. So whenever there is a presence of external ligands like glucagon, epinephrine, at that time it is going to bind with the GPCR, and that will result in the conformational change in the structure of a GPCR. Just after the binding of ligand to the receptor, the GEF, that is guanine nucleotide exchange factor, will come in action, and that will result in the replacement of a GDP to GTP, and this will result in the separation of a alpha subunit from the beta and gamma. Now the trimeric form has been degraded into a alpha monomeric form and beta and gamma dimer form, and here the GTP will remain attached to the alpha subunit, and after this the alpha subunit is going to do a further action or the downstream action. The next step is the activation of a adenylate cyclase enzyme that is present in the inner membrane of a plasma membrane. So, alpha subunit is going to activate this adenylate cyclase enzyme, and as a result, this enzyme is going to convert all the ATP that is present inside a cell into a cyclic AMP or cAMP. Now from this point, the cAMP or cyclic AMP dependent pathway will going to start. So in this pathway, there is an involvement of one enzyme, that is protein kinase A. So before moving to the further step, first of all we have to know the structure and the different role of the protein kinase A. So it is a family of enzyme, that is protein kinase A, whose activity depend upon the cellular level of a cAMP, and that is why this protein kinase A is also known as the cAMP dependent protein kinase. So in simple words, we can say that the activation of a protein kinase A will require the high level of cAMP inside a cell. Now let's talk about the structure of protein kinase A. So it has two regulatory subunit and two catalytic subunit. So in overall, it is going to make a tetramer structure. Now let's talk about the function of each subunit. The regulatory subunit used to regulate the function of a protein kinase A. That means whether this protein kinase A is going to activate or inactivate, that is going to be decided by the regulatory subunit. And if we talk about the catalytic subunit, it used to catalyze the biological actions. Whenever the regulatory and the catalytic subunit remain attached with each other, at that time the protein kinase A is mainly in an inactive form. So for activeness, this regulatory and catalytic subunit should detach or should separate it from each other. Now let's relate it with the pathway. So in the previous portion, we have seen that there was a secretion or there was a production of a cAMP with the help of a adenylate cyclase. Now what's happened? This cAMP or cyclic AMP has a site on the regulatory subunit of protein kinase A. So this cyclic AMP is going to bind to the specific location on the regulatory unit of the protein kinase, and that will going to cause a dissociation between the regulatory and the catalytic subunit. After dissociation, the catalytic subunit will become active, and now it is ready to enter inside the nucleus. Inside a nucleus, there is a transcription factor which is known as a CREB or CREB. The full form of CREB is a cAMP response element binding protein. Inside the nucleus, this CREB protein mainly remains in an inactive form. So for activation, it requires a phosphorylation, that means addition of a phosphate group in its structure. So that function, that is the phosphorylation, is mainly performed by the catalytic subunit because it used to transfer the phosphate from ATP to the specific serine and threonine residue of CREB protein. This thing you should remember, that the serine and threonine residue or the amino acid of CREB protein is going to participate in the phosphorylation. So in this way, we can say that the regulation of a transcription by the protein kinase A is mainly achieved by the direct phosphorylation of the transcription factor, that is CREB. After the activation of a CREB protein, it is going to take part in the stimulation of a different gene transcription. So if we summarize the whole pathway, so first of all, there is a need of an external ligand that is going to bind on the GPCR, and that will result in the activation of a G protein. Now this alpha GTP is having a capability to activate the adenylate cyclase enzyme, and this adenylate cyclase enzyme is going to convert the ATP into a cyclic AMP. Now the protein kinase A mainly requires the cyclic AMP for its activation. So that is why the high level of cAMP is going to bind to the regulatory subunit of a protein kinase A, and that will result in the dissociation of the regulatory subunits from catalytic subunit. Now this catalytic subunit is going to enter inside the nucleus, and here it is going to phosphorylate the CREB protein, and after the phosphorylation of a CREB protein, this CREB protein is going to activate, and then it is going to participate in the stimulation of a gene transcription. This pathway is very important from CSIR net exam point of view. I think in 2016 or in 2017, they have asked one question from this structure of a protein kinase A that mainly include the regulatory subunit and the catalytic subunit. So every step should be in your mind that why this particular step is taking place, what is the reason behind it, and how it is going to participate in the cellular response. One important tips I would like to give you, that don't go for the overview, because overview is only necessary during the revision time. But if you are preparing for any topic, just go in detail, because previously the CSIR net question was very easy and up to the average level. But nowadays the level of question paper is getting higher and higher day by day. So you should know the each and every detail of every path. So that is why I used to take a lot of time to explain you each and everything. The student who are seriously preparing for CSIR net or who have given 2 to 3 attempts for this examination, they know that there is no shortcut for clearing CSIR net. You have to study each and every point regarding the particular topic. Studying overview of any particular topic will not going to help you in solving the section C question, because that are mainly based on the concept. So if your concept is not clear, you will not be able to solve the section C question, because the memorizing or the overview is only going to help you in solving the section B question, and that also only for 10 to 15%. So first try to clear your whole concept regarding a particular topic, because in section C there are mainly the statement based question. You have to eliminate the wrong statement. So how will you able to eliminate that? If you are not knowing the concept or you are not knowing the particular pathway or each and every step of pathway, how you will going to eliminate the wrong statement from that question? So that is why I am saying that your concept should be clear. Go through each and every point. Then only you will going to perform very well in section C. Now the question is how this intracellular level of cAMP is regulated. So it is regulated by the balance between the activity of a two enzyme. The first enzyme is the adenylate cyclase and the second enzyme is the phosphodiesterase. We have already seen the activity of all these two enzymes in the previous slide. So the adenylate cyclase, it mainly used to convert ATP into cyclic AMP, and the function of a phosphodiesterase enzyme is that it used to dissociate or it used to degrade the cyclic AMP into a normal AMP. So in this way, the adenylate cyclase is going to perform its function in the synthesis of cyclic AMP, whereas phosphodiesterase enzyme, it used to help in the breakdown of cyclic AMP. So in this way, the regulation of the level of cAMP is mainly done. Now the question is that what is the significance of cAMP pathway? What is the importance of this pathway? Why we are studying this? It is not like just the pathway is given and we are studying. No, you should know the reason behind this. Why it is occurring? What is the reason behind this complicated pathway? So I have mentioned all the important points in very simple words. So we can read out from here that the protein kinase A enzyme is also known as the cAMP dependent enzyme, because it get activated only if cAMP is present, and that we have discussed in the previous slide. Once this pKa is activated, it phosphorylates a number of other proteins, including the enzyme that used to convert glycogen into glucose. So in the previous slide that was displaying the pathway of cyclic AMP, we have seen that the protein kinase A, it used to phosphorylate the CREP protein. So in this way only it used to phosphorylate many proteins that because it is involved in many different pathway. So first pathway that will include the conversion of a glycogen into the glucose, and in this pathway also the pKa is playing an important role. So after activation, the pKa may migrate to any other pathways like glycogen to glucose conversion. Second is the enzyme that promote the muscles contraction in the heart, leading to an increased heart rate. The third point, it will act as a transcription factor which regulates the gene expression. Not only pKa, this cyclic AMP is also going to play a different role in different location. Like many different cell response are mediated by the cyclic AMP. These include the increase in the heart rate, cortisol secretion, and breakdown of glycogen into fat. The cAMP is essential for maintenance of memory in the brain, relaxation in the heart, and water absorbed in the kidney. If cAMP dependent pathway is not controlled, it can ultimately lead to the hyperproliferation, which may contribute to the development and progression of cancer. So in this way, the cyclic AMP is going to play its role in different pathway that will include the conversion of a glycogen into glucose, the increase in the heart rate, and the expression of a particular gene. And along with that, it also help in the cortisol secretion, then breakdown of a glycogen. After that, it will going to help in the maintenance of memory in the brain, then relaxation of heart, water absorption in the kidney. So in this way, it is going to help in a biological process. Along with the pathway, we have studied the significance of the cAMP pathway too, because if there is a production of a cAMP and protein kinase A inside a cell, so not only in a cAMP pathway, it is going to perform in different pathways too, because it has the entry point so that it going to enter in some other pathway and it is going to perform a different function that we have mentioned in the significance point. Now we are going to start with the IP3-DAG pathway. This pathway will also include the receptor, that is mainly the GPCR receptor. The full form of IP3 is the Inositol triphosphate and full form of DAG is the diacylglycerol. Let's first talk about the ligand for this kind of a receptor that is present in a IP3-DAG pathway. So the ligand may be hormone, neurotransmitter, growth factor and any kind of a hypertrophic stimulus such as the angiotensin II. There are many different terms that we're going to include in this pathway. So first of all, we have to understand that term and then we are going to study about the pathway. So in diagram, first we can see the G protein coupled receptor, that is GPCR. So we are already familiar with what is GPCR. Then there is a presence of a ligand that is in the extracellular matrix, and we also know that what is ligand and what kind of ligand is going to bind to the GPCR, which is a class of a cell surface receptor. The next thing which you can see from the diagram is the PLC. So the full form of PLC is the phospholipase C. So first of all, the phospholipase C is a kind of an enzyme, and it is mainly the membrane associated enzyme that used to cleave the phospholipid just before the phosphate group. The next factor is the PIP2. The full form of PIP2 is the phosphatidyl inositol diphosphate, or you can say that the phosphatidyl inositol biphosphate. This PIP2 is a type of a phospholipid that is present in the plasma membrane. The next term is the IP3 and DAG. So this IP3 and DAG are very important secondary messenger molecules which is used in signal transduction and lipid signaling in biological cells. This DAG and IP3 mainly used to form whenever there is a degradation of a PIP2 or the membrane phospholipid PIP2 with the help of a phospholipase C. So before moving to the main pathway, the concept regarding the PLC, DAG, IP3 and secondary messenger should be clear, and I have tried my best to explain you all the concept regarding all these different terms. So now we are going to start with the IP3-DAG pathway. So first of all, I would like to mention that the PLC beta, PIP2, all these are membrane associated. PIP2 is a type of a phospholipid and PLC beta is a type of an enzyme that is associated in the plasma membrane. So first of all, there will be the presence of a extracellular ligand that is going to bind on the GPCR, and that will result in the conformational change in the structure of a GPCR. And that is going to result in the activation of a G protein. This activation will replace GDP with GTP. Now the alpha subunit of G protein interact with the membrane bound enzyme that is phospholipase C, and it will activate that enzyme. Now after the activation of a phospholipase C, it is going to catalyze the reaction in which it is going to break the PIP2 within a cell membrane, and after that the product of this breakdown will be the DAG and IP3. Here the phospholipase C or PLC which is going to act is mainly the phospholipase C beta, or you can say that PLC beta. After the formation of a DAG and IP3 from PIP2, the IP3 will going to migrate or diffuse in the cytosol, whereas DAG will remain in the plasma membrane due to its hydrophobic activity or property. Now this IP3 is having a receptor on the endoplasmic reticulum, and after binding to that receptor, the IP3 will lead to the opening of the calcium channel. In this way, the IP3 will stimulate the release of calcium ion from endoplasmic reticulum, whereas DAG is a physiological activator of a protein kinase C. The production of a DAG in the membrane facilitates the translocation of a PKC from cytosol to the plasma membrane. So initially this protein kinase C or PKC was present in the cytosol, but after the production of a DAG, it has been translocated from cytosol to the plasma membrane, that you can see in the figure. PKC or protein kinase C, it used to belong from the family of a kinase enzyme, and its function is to phosphorylate a particular target protein at a serine and threonine residue. In the cyclic AMP pathway, we have seen the similar function that was related to the protein kinase A, that it was also phosphorylating the target protein at a residue of serine and threonine, and protein kinase C also doing the same function, that is the phosphorylation of a target protein at serine and threonine amino acid residue. So in this way, the protein kinase C enzyme will become activated with the help of a calcium and DAG, because this protein kinase C enzyme has the affinity for the calcium ion. So the calcium ion positively regulate the protein kinase C, and in this way it will come in action and it is going to perform, or it will going to participate in the further cellular response. So PKC also activate its target protein by phosphorylating it. Let's summarize the whole pathway. So first of all, a ligand will come and bind to the GPCR, that will result in the activation of a G protein. After the activation of a G protein, the alpha subunit is going to activate the PLC beta, which is a phospholipase C enzyme that is associated with a plasma membrane. Now, the function of this PLC beta is to cleave the PIP2 phospholipid, and the product of this cleavage is going to be a DAG and a IP3. DAG will remain attached to the plasma membrane because it has a hydrophobic nature, whereas IP3, it will going to migrate in a cytosol. Now, the ER is having a receptor for the IP3. So, when IP3 is going to bind to that receptor, that will lead to the opening of a calcium channel, and there is a release of calcium outside the ER. Now, this calcium is having an affinity to the PKC. So, initially the PKC was present in the cytosol, but with the action of a DAG, it has been came in contact with the plasma membrane. Now, the calcium will come and binds to the PKC, and as a result, this PKC will get activated, and now this PKC is ready to phosphorylate its target protein and do a cellular response. Now we are going to talk about the significance of IP3-DAG pathway. So we have seen that there is a release of a calcium ion in a very large amount, as well as there is activation of a PKC, that is a protein kinase C. So obviously they are going to enter in some other pathways too. So in this way we are going to see that what is the importance or the significance of this pathway. IP3 has main function to mobilize the calcium from storage organelles and to regulate the cell proliferation and other cellular reaction that requires the free calcium. So in simple words, we can say that the cellular need of calcium by different organelles or by different chemical reaction is mainly going to occur or completed with the help of a IP3, because it is only the factor that is going to make calcium available for the different reaction. Along with that, the IP3 is going to serve as a second messenger for a nervous system. The IP3-DAG pathway is also having a very important role in the slow block of polyspermy in a sea urchin. So it is a particular creature that is present in a water. So when we are going to study about the sea urchin in our developmental biology, at that time we will see that there is a slow blockage of polyspermy with the help of this IP3-DAG pathway, because the high amount of a calcium that is going to release from this pathway, that is going to help in the polyspermy blockage. So these are some important points that is related or that is in favor of the significance of IP3-DAG pathway. You will get many more different points and especially try to find that, so that you will be able to relate this pathway with the particular examples. I have written all these steps of IP3-DAG pathway. If you want to read, you can pause the video and you can read each line, although I have explained each and every point that has been present here with the help of a diagram. These are the definitions of different factors that we have studied in all these two pathways. If you want to study or if you want to know more, you can take a help from these slides. Although I have kept it so that it might be helpful for you if you want to know more about all these factors. During the explanation, I have explained each and every factors that I have kept in this slide. So with this, we have completed the cAMP pathway and the IP3-DAG pathway. So try to solve different questions on these two topics so that you will be able to relate all of your knowledge according to the question. If you are new to my channel, you can subscribe my channel for getting more different videos on CSIR, NET, GATE and many more different examination. Along with that, don't forget to hit the bell icon so that you will be notified with my latest video uploads.